So today we'll focus on microbial interactions with humans, epidemiology, and transmission of disease. So a lot of the stuff that we talk about today is quite applicable to your real life situations. The first thing we have with regard to mechanisms of pathogenicity is a slide that you've seen before. Now remember I said you have to know the 5 requirements for a microbe to be pathogenic, to have a successful infection. So for a microbe to be pathogenic, it must be able to enter the body or cell, stay in, defeat your immune system, your defenses, cause some type of damage, and transmit to a new host. So some students remember it as ESDDT. Enter, stay in, defeat, do damage, and transmit. Now what's new on this slide now is the term virulence factors, and I want you to circle star, highlight that term, because virulence factors is something that will come up multiple times throughout this course, and you have to know the definition. What virulence factors means is any molecules that are produced or secreted by the bacteria to do all of these above things. Okay, so virulence factors are any molecules or structures that help bacteria do any of these 5 requirements for pathogenicity. Okay, one more time. Virulence factors are any molecules or structures that let bacteria or help bacteria do any of these 5 things to be pathogenic. So we're going to go one by one through these various requirements for successful infection. The first requirement for a microbe to be pathogenic is to be able to enter the body or the cell. There are various ways that they can enter the cell or the body. Okay? We call that portal of entry, meaning how it's entering, where the microbe's actually getting in. And a lot of times this portal of entry can also serve as an exit as well. So there are 2 main categories of portals of entries for bacteria. The first ones are mucous membranes, okay? So any of the tracts that you see listed here, such as respiratory, gastro, genitourinary, conjunctiva, all of these have holes adjacent to the external environment and allow for microbes to get in. So when you look at these tracts, there are a few things you have to ask yourself. First of all, which of these tracts is going to be the most common and most problematic portal of entry? Well, that would be the respiratory tract, because think of it, you always have to breathe all day, every day. The gastrointestinal tract will be the second most problematic portal of entry because you always have to eat or drink. But at least with that one, it's not every second of the day, the way breathing is. Genitourinary is becoming more and more problematic as a portal of entry, and we'll talk more about that in the STD lesson. But for now, if you think about it, it's not, even though it's very, very high rates and problems today, when you look at it compared to the other tracts, people don't have to have sex 24/7 the way they have to breathe. So it's not as common as respiratory, taking in microbes. The last one is conjunctiva, and conjunctiva, right next to that, write the word eyes. Okay, conjunctiva means your eyes. And we'll go through in a later lesson how your eyes actually do a lot of things to prevent microbes getting in this portal of entry. Okay? The other big portal of entry is the skin. And any bacteria that enters through the skin, through breaks in the skin, is considered as entering through the parenteral route. Now, when we think of the skin, remember the skin is the largest organ of the body. And we're going to talk about the skin and the immune system as being very good at protecting us. But even though it's very good at protecting us, a lot of microbes end up penetrating the skin barrier and entering through the parenteral route entry. And again, parenteral route simply means that a microbe enters your body through a break in the skin. So examples of that would be any kind of cut, puncture, injections, having surgery, or even things like insect or dog bites, all of which we'll go through in various lectures throughout this course. The big thing is make sure you know terminology, what it means to be a portal of entry, what the examples of portals of entry are, and what the term parenteral route means. So once a microbe has entered our body, a lot of them now want to try and enter our cells. But if you think about it, each of your cells has that plasma membrane, which is like a barrier, kind of helping to block the pathogen. But many of the pathogens have ways to then break through this barrier and make it. So what a lot of them do, especially viruses, is they use specific receptors that are on the outside of your cells. And what they can do is they can then basically bind to that cell, merge with that outer membrane, and then insert themselves inside of the cell. Kind of like you see in this figure here, where the virus has bound to the receptor and then merged with the membrane and popped itself right inside of the human cell. What they then do is when they break free of that cell later on, they burst through or lyse that cell and they steal part of that cell's membrane. And they basically wrap themselves with a piece of the host cell, meaning you, your cell's membrane around it to then travel through your body. And you kind of ask yourself, well, why would a virus want to do that? Why would a virus want to wrap itself in this extra envelope that's basically part of our cells? Well, think about it. What's on that outer envelope? What did we say was on there? Receptors. So now that virus can travel around in your body, basically wearing a coat that looks like one of your cells. Match your cells, which makes it a lot easier for that virus to escape your immune system and also to infect other cells within you. So if you ever see a virus that has an envelope, because we're going to mention them in later lectures, that means that it stole that membrane from one of the host cells. Basically, we say that viruses are jerks. Now, once the pathogen has entered your body or your cells, what's the next step that it has to do to be a true pathogen? Well, it has to stay in. Because think about it, if you instantly coughed or sneezed it out, it's not going to do any damage. It's not going to be a problem or a pathogen. So we kind of touched upon some of the structures that certain bacteria or microbes have that help them to stay in. Okay, so we mentioned fimbriae, which are these hair-like extensions here, which help them to stick to things such as your cells or tissues. A lot of bacteria also have what's called adhesins. And adhesins, which you see in the figure here, they're simply surface proteins, basically receptors, that help them grab a hold of receptors or proteins that are on your cells or tissues. A big example of this adherence using adhesins are the bacteria that make up plaque on your teeth. Okay, they also tend to have glycocalyx, which we mentioned was that sticky capsule that also helps them to adhere to things. So just to kind of recap one of the big terms that I said you would have to remember, if we say that fimbriae and adhesins or the glycocalyx, any of these structures are meant to help the bacteria stay in, which is one of those 5 characteristics of a pathogen, then what do we call them? They're virulence factors. Okay, so remember the term virulence factors. Fimbriae, adhesins, glycocalyx, any of those are examples of virulence factors. They help the microbe to be a pathogen. Now, when we talk about staying in, another example of how bacteria can stay in is the example of spirochetes, such as Treponema, which is one of my favorite of the giant plushies that you'll see that are present in the lab. Treponema is spirochete, which is why it has that spiral shape. It's what causes syphilis, which we'll talk about in more detail in the STD lesson. But what's important about this guy here, syphilis or Treponema, is that it uses axial filaments, which we mentioned in an earlier slide. The axial filaments let the Treponema, or causing syphilis, embed itself or corkscrew into the human's tissues and organs to really attach, to stay in. Okay, so whenever you hear syphilis, think of axial filaments. And that's how it is motile, and that's how it establishes or attaches to different tissues. Now, when we talk about microbes attaching, adhering, getting established within the human body, the number of microbes that actually establish themselves determines how successful an infection is present. Now, the number required for this kind of success varies, and there's two terms that I want you to be familiar with, especially if you go into the medical field, because you'll notice in case studies or various documents, you'll frequently see the term LD50 or ID50. What these are lethal dose 50% and infectious dose 50%, okay? So we'll first give the straight, textbook kind of definitions, but then I'll kind of give you a little insight into how to really work with these values. So the official definition for lethal dose 50% or LD50 is the number of organisms needed to kill 50% of the host. Infectious dose 50%, on the other hand, is the number of organisms, meaning pathogens, needed for 50% of the population to show symptoms, okay? So now the way to really understand these terms is to think about what it means if you have a low version of the term or a high version of the term. So for instance, if for a particular bacteria, let's say, we just mentioned syphilis, let's say we were told that syphilis has a low LD50 and a low ID50, what would that tell you about the syphilis pathogen? Well, if it's low LD50 and low ID50, that means it only takes a couple of this bacteria, picture, one or two of this bacteria and boom, half the people die. Or one or two of this bacteria and boom, right away 50% is showing major symptoms, brain dysfunction and severe illness. Well, if it only takes a couple of them, a low number of them, to make people so sick, well, that tells you that each individual one must be really, really strong, right? Whereas if there's a high LD50 or a high ID50, what would that tell you? Well, you need millions of this little bacteria before people even sniff them. Or you need millions of these just to kill one person. Okay? That would tell you that each individual one isn't very strong, right? So for the first question that I have highlighted in red, this one here, low LD50 and or a low ID50 tells you the pathogen is very virulent. Okay, so for this one, write very virulent. Whereas a high value would tell you not very virulent. Okay, low virulence, not a strong pathogen. Okay, so make sure you're very comfortable with those two terms, and if you need a little clarification, just send me a remind message or an email. Now, once the pathogen has entered and stayed in, the next thing it must do is defeat the defenses. And remember, we said that basically means defeating whatever your immune system is throwing at it to try and kill it. Now, when we talk about defeating the defenses, you have to know the difference between passive and active strategies. So if something is passive, remember, active means energy, whereas passive means no energy. Very passive, you don't need to do anything extra. It's just there, right? So for bacteria, if they're trying to defeat the host defenses passively, it tells you that it's something about them that they don't have to do anything extra for. They already have it. So passive would be built-in structures on the bacteria to protect them, such as capsules, which can physically block what you're throwing at it, or help prevent any of your immune system cells from eating them up, which is phagocytosis, or their cell wall, which again physically blocks your immune systems targeting them, but also has proteins that helps them to do what they need to do in your body, and that can be toxic or dangerous to your body to help protect them further. Okay? So again, passive strategies are parts of the bacteria that they already have to protect them. They don't have to do anything extra. Whereas active strategy, if you are active, you've got a lot of energy, right? You got a lot of movement. So for active strategies, now the bacteria is going to be doing something active. It's going to be producing something, secreting something. You got movement here, right? So active strategy would be any of the enzymes that they produce and release to kill off your immune cells. The three big examples of active strategy are leukocidins, hemolysins, and coagulase. So leukocidins, that's an enzyme that will kill leukocytes, which means white blood cells. Hemolysins instead, hemo, are now going to kill red blood cells, causing those red blood cells to lyse. And then coagulase, I'm sure you've all heard the term coagulating. Coagulase will cause blood clots in the host, which slows down those immune cells from getting around. Okay, so make sure you're very comfortable with the term passive versus active and what the examples are of both of these. This slide here is just to show you a picture of what we were just talking about in terms of passive. So passive strategies having the capsule or the cell wall around the bacteria, okay? And active being anything getting secreted in any of these, okay? But again, you never have to memorize any of these figures. They're just to help you understand or visualize what we were just talking about. Now, the last thing that I want to mention over here is the idea of quorum sensing and biofilms. Okay, both of these play a role in pathogen virulence, being that quorum sensing, if you picture this, what this is proteins basically will not be activated and released unless the bacteria assesses the population around it and determines, hey, I have a lot of my buddies around, so now let me activate these genes and produce these proteins and these virulence factors. Now, why would a bacteria want to wait until it senses the environment around it and knows that there are a lot of other bacteria around it before it starts activating genes and producing proteins? Well, think about it. When we went over genetics and gene expression and translation after that, it's a lot of work. There's a lot of enzymes involved, a lot of steps involved. It takes a lot for a bacteria to do these things. So why would you, if you're a bacteria, waste your resources, your energy, your enzymes, and honestly your time producing these proteins and these virulence factors if you're just one little bacteria and you're not going to be able to do much with that? Why not wait until you have a whole big population around you a whole bunch of bacteria friends, where now if you all use up that energy and resources to produce the virulence factors, now it'll do something. Now the host will actually feel it and you'll accomplish something. Okay, so quorum sensing, the reason they do that is to conserve resources until they know that there are other bacteria around to help complete the task of hurting the host. In addition to quorum sensing, another thing bacteria can do in order to help with being pathogenic and virulence is to produce biofilms. Okay, so certain bacteria will produce biofilms. What this means is they will form a collection or an aggregation of different species of bacteria that stick together and will grow as one big mass. Okay, they're all working together, sticking together. Now, if you think about this, this is kind of valuable to these bacteria because now you have a whole bunch of species of bacteria with you. Okay, so you can obtain different nutrients, you can get different things done, and most importantly, when the host tries to damage you or destroy you, well, if you think about it, a lot of the things that we do to destroy bacteria, it'll get rid of one type, two types, but it's not going to get rid of every single possible type at once. So by having a variety of different species working together, they increase their odds of survival because you can't kill them all at once. So biofilms become very tricky for us to destroy or clean up. And some of the big examples of that, the one that you will constantly hear this semester, and I want you to remember, is plaque. Plaque on your teeth is a biofilm. And that's why it's so difficult to deal with, and that's why you'll have plaque for your whole life. You can never 100% get rid of it. You can minimize it, but you can't get rid of it. Now, the other area that we commonly see biofilms is on medical devices, especially things like catheters or equipment, medical equipment. Okay, and that's why you always have to be very careful in the medical field and try and minimize the chances of any biofilm collection. You also see biofilms in things like pipes and whatnot and sewage, but the big examples that I want you to remember for our purposes are plaque on the teeth and biofilms on medical equipment, such as catheters or IVs. Now, the last of the things we're going to mention today is how pathogens will try to damage the host. Okay, damaging the host. Now, with damaging the host, there are two big causes. The damage can either be because of the pathogen itself being present and active, or the damage to your body can actually be because of your own body, because your immune system is trying to fight those bacterial cells or those viruses and accidentally, friendly fire, injure your own cells or damage your own cells and tissues. Okay, so if you think about things like excess inflammation, your immune system's trying to help you, but it can also be quite problematic if you have too much inflammation. So when we talk about damaging the host, there are two big categories to remember. There's direct damage or indirect damage when we're talking about damage being caused by the pathogen itself. Now, if it's direct damage, then you're basically going to think of it as the direct damage from the physical pathogen itself. So kind of picture if that pathogen is kind of crashing into cells and tissues. So the pathogen itself is destroying host cells or tissues. Okay? Indirect damage is now when you have a systemic, so body-wide trouble because of toxins produced by the pathogens. And in the next couple of slides, we're going to talk about the two big categories of toxins, and you have to remember these toxins, okay? So you are going to circle, star, highlight toxins, okay? So the two categories of toxins, again, putting stars all over this slide and the next slide, the two big categories of toxins are exotoxins and endotoxins. So when you hear the word exo like exoskeleton, that tells you it's on the outside. It's released. So exotoxins are secreted to the outside of the cell, of the bacterial cell. The fact that it's secreted and it's from living bacteria, this is very problematic to us, and it makes them the most lethal of toxins. Okay, so circle the fact that these are the most lethal and put a little star next to living bacteria secreting. Okay, the reason these are most lethal is because think about it. These toxins can act remotely without the pathogen, and if you kill the bacteria, that's not going to be enough to heal the patient. You still have these toxins floating around through their body. Now, what is most lethal about them, though, is the fact that these are toxins being secreted by living bacteria. They are still alive, which means they can continuously pump more and more and more toxin throughout your body. That's more lethal and damaging to you because it's a factory, basically, consistently pumping this toxin throughout your body. You'll see in a minute why this or how this varies from endotoxins, which are not as toxic. Now, the big examples of exotoxins that you tend to encounter are cytotoxins, neurotoxins, and enterotoxins. Whenever you hear the term cyto in biology, cyto means cells. So cytotoxins will kill any cell that it comes in contact with. A big example of this is anthrax. So if you heard all those times there were anthrax scares, that's because the anthrax toxin will be cytotoxic and it will cause major hemorrhages and pulmonary edema as it's destroying cell after cell. Neurotoxins, another example of exotoxins, are also very lethal because these guys interfere with your neurological signals. So a big example of this that I want you to remember is botulinum toxin, so the toxin from botulism, and remember tetanus. So botulism and tetanus will produce neurotoxins. Since they're targeting the neurological signals in your body, that means that they'll block things like acetylcholine and cause paralysis. Now you may ask yourself, well, how is paralysis lethal? Well, it's not just muscles, regular old moving your arms and legs that becomes paralyzed. What other muscles can become paralyzed? Cardiac and respiratory. So you lose the ability to pump blood or to breathe, so the patient dies. So neurotoxins are usually rather fast-acting and dangerous. The last one on the list here for exotoxins are enterotoxins. And whenever you see the word entero or enteric, remember GI tract. So enterotoxins will affect the lining of the digestive system and cause things like diarrhea and vomiting, which can be dangerous in terms of dehydration. Now, the alternative is some bacteria instead will produce endotoxins. And when you hear endo means within. So endotoxins are within the bacteria's own cell wall. They're in the outer layers of gram-negative cells usually. So for instance, the lipids that we mentioned in earlier slides. The difference with endotoxins, since they're built into the bacteria's cell wall, endotoxins are only released when the cell dies. This means that they're not as toxic as exotoxins, because think of it, what's more dangerous if you have a serial killer or if you have someone who commits suicide when they commit the murder? Well, the suicide killer, they only can kill that whoever they killed at that moment. They can't kill anymore once they're dead. Whereas a serial killer, which is the exotoxins, they're still alive. They can keep killing, keep releasing toxins. So endotoxins are not as toxic because they're basically suicide death. Okay? So endotoxins are less toxic and released only when they die. And the other thing that they're most known for is causing fever. Okay, fever. Now, one last bullet point that I mentioned here is that endotoxins are more heat stable. And that makes sense since we just said that they target fever, so it makes sense that they're able to withstand the heat. It's basically because these are lipopolysaccharides usually, whereas exotoxins are soluble proteins that can end up denaturing. Okay? So the last thing I want to show you is when we say trigger fever, this is what we actually mean. So with endotoxins, what happens is you have a bacteria here, and it gets engulfed by one of your immune cells, a macrophage that's known for phagocytosis, which is trying to engulf and break down the bacteria. Your immune cell thinks that it's protecting you and doing good by breaking down this bacteria and killing it for you. What it doesn't realize is that this bacteria, usually a gram-negative cell, once broken down, as you can see here, the little pieces of the outer membranes break apart, break down, and those happen to be endotoxins. These endotoxins then induce cytokines, which are these little chemical circles here, and the cytokines then travel and activate the hypothalamus. The hypothalamus will produce prostaglandins, and that's basically the thermostat in your body and causes your temperature to increase, okay, to give you a fever. A fever is actually part of your immune system, and fever is meant to do good for you. We're going to talk about this in a later lecture, but for now, just ask yourself, well, why would a really high temperature be good? Well, bacteria, they're not going to like that high temperature. So it's trying to help reduce how favorable your body is for the bacteria. Now, again, this is just to show you kind of how endotoxins work, but you don't have to memorize this figure. Just be aware that endotoxins are the type of toxin that will induce fever and that are not as lethal usually as exotoxins. I really want you to remember exotoxins as the big bad guys. Bad like an X. So like I said, today there is a lot of terminology, and so the first bit of terminology in the field of diseases that we need to kind of distinguish amongst includes the terms pathology, etiology, and pathogenesis. And these each have their kind of distinguishing factors about what they truly mean. The first one is pathology, and as you can see from the beginning of the word, patho, this one is dealing with pathogens. Specifically, pathology means the study of disease. The second word here, etiology, now we're looking at the cause of a disease. So for instance, trying to identify the exact bacteria that causes a particular infection. The next term is pathogenesis. Now, patho you saw as disease in the first term, but now when you include genesis, anything that involves genesis talks about the beginning of things. So pathogenesis is defined as studying the development or beginning of a particular disease. Okay, so again, pathology is studying disease in general. Etiology is studying the cause of a disease, such as the exact bacteria or the exact virus. And pathogenesis is studying the development or progression of that disease. The next two terms that you should be able to distinguish, even though you hear them all the time, you may currently kind of mix and match their meanings or use them interchangeably when they're actually quite different from each other. These two terms are infection and disease. Infection is the actual invasion or colonization of your body by the pathogens, so by the bacteria or the viruses or the fungi. Okay, so it's basically the pathogens taking over your body and being present. Disease then means the abnormal state that we observe with your body after it's been colonized or due to that colonization. Okay, So once again, infection means the invasion or colonization of your body by pathogens, whereas disease is the abnormal state the body ends up in due to that colonization. Okay, basically the actual feeling sick part. Okay. So you'll notice that since today's lesson involves a lot of terminology, I've added some slides for you to be able to write down or recap some of the information that I mentioned. Okay, I purposely did not put these in the PDFs that I gave you because, well, you have to actually listen to the lectures to get all of the information. Okay, so now this is to recap each of the terms that we just talked about, so you can pause the recording in order to write down all of these terms. Remember, terminology is very easy to be asked as multiple choice questions on an exam. Okay? Now, when we talk about diseases, infections, and all of this fun stuff, there is a reminder I want to make. Not all microbes are going to be causing infections. Not all microbes are pathogens. Okay? Remember that there is a lot of microbes on or in your body that are not doing you harm under normal circumstances. And what do we call those guys? Your normal flora. Okay, so what do we call the harmless ones in or on our body? The answer to that is your normal flora. Now, with all of those microbes in and on your body, there's another term that you sometimes hear, which is the microbiome, which is becoming a big topic in terms of research and publications. A lot of scientists are studying and understanding the microbiome for various purposes. What the microbiome actually means is all of the microbes, so that includes their DNA and their physical structures, as well as the environment that they're all interacting on. So for instance, one of the big things to study is the human microbiome. This is basically studying all of the microbes that are all over your body, as well as your body itself, because that's the environment that they're interacting with. Okay, we could gain a lot of information from that. And the last thing I want to point out on this slide is there are various things that can influence the microbiome, especially the human microbiome. So basically, the normal flora that you have on and in your body, they're always slightly changing, okay, from day to day, morning till night. Things such as your diet, your activity, were you very active? Were you sweating a lot and thus changing the pH of your skin? Did you go too long without eating? Or did you eat a bunch of salty things or a bunch of sugary things? Or did you drink a whole lot of alcohol? Hey, not judging here, because all of those things can affect whether or not the microbes want to be in that particular location and how well they survive. Also, even things such as your gender can affect your microbiome, okay? Because for instance, if you think about the various genders that exist, your bodies have different hormones excreted. So for instance, if we have a gender releasing a whole lot of testosterone, okay, that can affect the microbes that want to be present at a particular time. Okay, so there's a lot of different things that can affect your microbiome. Now, speaking of normal flora, this is something that we've mentioned quite a few times throughout the semester. We've been really focusing so far on the fact of normal flora being the good guys, all over your body, helping you out. But we do want to remind ourselves that even the good guys can be opportunistic pathogens. Okay, so what does it mean to be an opportunistic pathogen? Again, those are the bacteria or the microbes where normally they're being good, they're helping us. But if the opportunity presents itself, they will take advantage. Okay, so the two opportunities that I keep reminding you about are if you become immunocompromised or if the microbe ends up in the wrong place. So for instance, E. coli that's supposed to be in your digestive tract, if it suddenly ends up in your genitourinary tract, boom, you have an infection and it becomes pathogenic. Okay, so that's what we mean by normal flora being opportunistic pathogens. Now, let's get back to the fact that they do normally help us and protect us. And the exact way that they do that is through microbial antagonism. Okay, so if you antagonize something, you're going against it, right? So normal flora can go against other microbes. Now, the way that they're protecting us through this antagonism, first of all, they're competitively inhibiting the growth of other pathogens, of other microbes. Because think about it, if the normal flora is taking up that space and using up those resources, there's not going to be any left, or not going to be much left, for pathogens to take advantage of, to flourish. Okay, so by simply just sitting on your skin, they're helping you out by taking up that space so that the bad guys won't sit there and start replicating. Some of them also produce bacteriocins, and that ending sins is to show that they're toxins, but specifically to other bacteria. So bacteriocins are toxins that will kill bacteria, and what the normal flora do is they will have toxins that kill the most closely related bacteria. Now, I know you might hear that and think, wow, what jerks, why do they want to kill their relatives? Well, because think about it, the more closely related those other bacteria are, the more similar their needs are going to be, right? They're going to want the same resources, the same food, the same space. And we just mentioned competitive nature. Bacteria is not going to want that to happen. So that's why they tend to produce toxins that kill the more closely related bacteria, because those are their biggest competitors. In addition to that, just the fact of them, eating and excreting their metabolic waste products all affect the area that they're living in, which affects which other microbes would want to be there. So this last bullet point here is pointing out that the fact that these normal flora do things like affect pH, because think about it, I know you don't want to think about it, but they are technically excreting all over your body. So you got basically the bacteria version of urine and poop all over you. That will change the pH of that location. And as we've seen throughout this course, other bacteria, each little bacteria has its own preferred pH, own preferred kind of, environment that it wants. So by affecting pH and even things like the available oxygen, they're reducing the likelihood of other microbes being present. Now, this slide is a reminder of the 3 possible relationships that can exist between hosts and microbes. These are terms that I'm sure you've encountered in other courses throughout your educational career, but just a reminder, when we talk about relationships between organisms, okay? I know it sounds very romantic, but instead we're talking about symbiosis, okay? Symbiotic relationships are the interactions between 2 organisms. The 3 examples that we have here are commensalism, mutualism, and parasitism. So the easiest one that students tend to remember first is mutualism, because if something is mutual, both parties will benefit. So that's the example in the middle here. Mutualism is when both organisms interacting get a benefit, such as the E. coli living in our digestive tract. Okay, because they get tons of yummy food and good space to live in. And at the same time, their enzymes help us digest things that we wouldn't have been able to digest, such as cellulose of plant products. Okay, so you wouldn't be able to digest vegetables without their help. The next one that students find it easy to remember is parasitism over here. Because when you think of parasites, what do you think of? Well, I think of undergrads, but most people think of anything bad, right? Anything feeding off of you. taking your energy, taking your nutrients, just taking, and not really helping in any way, right? So in this case, parasitism means one of the organisms, so the parasite, benefits, but at the expense of the host, okay? So the host gets harmed or used up in this relationship. The example that we have here is H1N1. And if any of you remember H1N1 from the news quite a few years ago, H1N1 is swine flu. Okay, so any of the things like flus or colds, the things making you really sick, that'll be parasitism. Now, the term that students tend to forget is commensalism. Okay, so we've discussed so far when one of the organisms benefits, when one of the organisms is harmed. So commensalism is when the host is unaffected. Okay, so you don't even realize that this microbe is present. It's not helping you, it's not hurting you. Okay, so that's commensalism, where the microbe will benefit, such as by getting food or space, whereas the host, completely unaffected, not helped, not harmed. Now, in this figure, they give the example of staph epidermidis on your skin, but I don't like that example, because if you think of any of the microbes living on your skin, what did we say about them? They're technically helping you out, right? That staph epidermidis is taking up space, taking up resources, so that the pathogens won't be able to live there. Okay, so I don't really like that example, but they use it because there are very few commensalism examples, okay, especially with regard to humans. You usually instead find commensalism as fungi living out in nature, okay, and we'll talk more about that in another lecture. The next concept to go over is etiology, which a few slides ago we defined as trying to study the cause of a disease. Now, there's a terminology that they use in this process, which is called Koch's postulates. So whenever you hear or see the word Koch's postulates, understand that this is a process to identify or pinpoint if a particular bacteria is causing a particular disease. Okay, so if you want to know for sure that this particular bacteria or virus causes this particular disease, then you would use Koch's postulates. So I listed them out on this slide, but in the next figure, we're going to get to see them visually. Now, when you look at this, there are four main aspects of Koch's postulates to prove or pinpoint the cause of a disease. The first one makes very good sense. The same pathogen has to be present in every case of the disease, because think about it. If someone has disease A, but doesn't have the pathogen that you're suspecting of causing disease A, well then how could that pathogen be the cause if someone has that disease without that pathogen present? Okay, so the first aspect of Koch's postulates is that the same pathogen must be present in every case. For the rest of these, I'm going to move on to the next slide so you can visualize them at the same time. So like I said, this is the visualization. After we know that the same pathogen is present in every case of the disease, then what we want to do here with Koch's postulates is you isolate or extract the suspected microbe from a diseased or dead animal, and you culture it. You then take that microbe and you inject it into a healthy animal and look to see, does that healthy animal then exhibit the same disease that we were studying? If they exhibit it, you then extract the microbe and study it to see, is that microbe the same one that you isolated from the initial dead or diseased organism? Okay, so again, what Koch's postulates is doing, you take a microbe you isolate from the diseased animal, you culture it and put it in a healthy animal, and if it is the true cause of that disease, it should then make the healthy animal show the same symptoms, the same disease as the initial animal. You can then extract it and study it to confirm that it even looks the same and is the same microbe. Okay? So once again, whenever you hear Koch's postulates, remember that the purpose is etiology, to pinpoint the exact cause of a disease, the exact microorganism causing that particular disease. Now, when we talk about these various infectious diseases, there's some terminology that you'll come across. This is another one of those slides where I say, I'm sure at one point or another, you've all used these terms or heard these terms, but you may not have known the true distinguishing aspects of the terms, or even if you were using them properly. So, the first term is symptoms. And your symptoms are the changes in your body function that are felt by a patient as a result of disease, meaning all of the things that you can visually see and feel due to having an infectious disease. Then we get to the next terms, and these terms are communicable, contagious, and non-communicable. Okay, all three describe infectious diseases, and you have to know the difference amongst them, as well as the examples of them. So first up, we have communicable diseases. Communicable diseases means that the disease can be spread from one host to another, but that it won't be that easily or quickly spread. So, for instance, the way to better understand this is to think of the examples. So the examples I like to give for communicable diseases are food poisoning and STDs. As you know, these can be caught from other people, but you won't catch it just by standing next to someone who's sick with one of these types of infectious diseases. Okay, so just by sitting next to someone who has an STD, you won't catch that STD. And as a New Yorker, I'm very grateful for that, because traveling on public transportation, everyone would have an STD just by sitting and standing on those buses and subways. Now, when we then talk about contagious diseases, now it's a disease that's easily and rapidly spread from one host to another. So now with contagious diseases, just by sitting or standing in the same room as someone who has this infection, you can get it, okay? In fact, they might have been in that room a few minutes before you and left by the time you got there, and you can get it. So for instance, just sitting in a seat in a food court, okay? Maybe someone who had the flu was sitting there right before you, sneezed all over that table, those chairs and all. It's all in the air, it's on the surfaces, and you can now catch it. Okay, so these are contagious diseases, things like the plague, like the flu, even like the common cold, where you can catch it very easily. And you notice with these, if someone in your family catches the flu, odds are you're going to catch it too, just by being in the same house as them. Okay, that is contagious. The last one is non-communicable diseases. Okay, so this disease cannot be spread from one host to another. So these are diseases you cannot catch from another person. Instead, you're catching them from things like soil. Okay, so the example of non-communicable diseases is tetanus. Tetanus, you can't catch tetanus from another person. Instead, you catch it from spores in soil, okay, so from inanimate objects. Now, just a reminder, as I say the word tetanus, anytime you hear tetanus, I want you to think tetanus and botulism are both two very problematic infections because of exotoxins, okay, very dangerous infections because of the neurotoxins that they produce, which are exotoxins, okay. Just in case I spoke too quickly during the last slide, this is a recap slide so that you have a chance to pause the recording and write down what I wrote or what I said for each of the definitions that you need to be able to distinguish. And again, make sure you're always comfortable knowing the examples as well as the terminology. So now, like I said, this lecture is a whole lot of terminology, okay? And again, they're terms that I'm sure you've encountered before, but maybe didn't know the true usage of them, okay? So here we have terminology relating to controlling the spread of disease. So the terms we have are isolation, quarantine, and vector control. When we talk about isolation, I always have students circle or underline that I, that first I of isolation. So when you hear isolation, think I or Roman numeral 1, okay? Because in isolation, you are only separating the sick individual, just the sick person. Whereas in quarantine, you can circle or underline the first 3 letters, okay? Quarantine starts with qua, just like quantity, okay? Because a quarantine, you are now separating a quantity of people. You are separating all of the healthy individuals who had contact with the sick person and may now be infected, but in the incubation stage and not showing symptoms. Okay, so the example would be if you picture a classroom. If student A is sick and has an infection and we isolate or separate student A from the rest of the class, that would be isolation. What we would then do is take all of the other students in the class and separate them from the public just in case any of them have caught this infection, because you don't want them running around, you know, going on the bus, going on the subway, going into stores or the gym or other classrooms, and now spreading this infection, which honestly, the concept of quarantine is very difficult to do. It's very difficult to control because, again, so many people don't realize when they are carrying around an infection before they start to show symptoms, okay, which we'll get to in the next page or the next slide. The last one here is vector control. What vector control is that you destroy the habitats or food or other resources that vectors rely on. And what we mean by vector are bugs, things like arthropods, things like mosquitoes. So the example of vector control that'll help you remember what we mean by this is the use of screens or netting. So think of mosquito netting and screens, or even all of the mosquito spraying that you saw a few years ago to reduce the risk of things like West Nile or Zika. So now, once again, I've wrote a slide of some recap notes just in case you missed any of the information that I was saying on the last slide. So again, feel free to pause the video right now just so that you make sure to write down everything here on this slide. Now we have a very interesting figure for you to know. Okay, so normally you don't have to memorize any figures, but this figure you need to know each of these stages. This figure represents the development of a disease, the 5 stages of infection that you will go through no matter which infection you are suffering from. And yes, sometimes certain stages within this cycle will be less visual or less noticeable, maybe last a shorter time or a longer time. But no matter which infection you have, you will go through all of these stages, if you make it through that infection, that is. Okay? Now, when you go through this, I want you to not only be aware of the name of each stage, but also the key characteristics of that stage. So for instance, when we look at this graph, over here, when we see number of microbes, you can write severity. Okay, so this axis represents the severity of your symptoms and of that infection. Okay, so the higher we are, the worse the symptoms and infection will be at that point. Now, going stage by stage, the first stage of any infection is the incubation period that we were just talking about in the previous slides. This is the time when you have the pathogen in your body, but you don't realize it yet because there are no signs or symptoms. At this point, the bacteria or the virus is just settling in. It's going through those initial stages of the pathogen cycle, which we mentioned as attachment. So it's trying to really embed itself, and it's starting to replicate or prepare enzymes, okay? But it's not reproducing itself yet. You then head into the prodromal period, okay? Prodromal period is the second period of infection. What this means is you are now exhibiting the mild signs or symptoms. So what I want you to write for the prodromal stage or prodromal period is mild general symptoms. Okay, whenever you see prodromal, I want you to think general symptoms. And if you think about any time you've just started to get sick, where you're like, oh, this is gonna be bad, I know it, it's usually when you get those general symptoms like malaise, okay, the really bad fatigue, or as I call it, that yucky feeling, right before you start to get really sick. So no matter which infection you have, during prodromal period, you'll have general symptoms that match any infection. So the big one being malaise or fatigue. The third stage in infection is the period of illness. Now for this one, I want you to write specific symptoms. Okay, so in prodromal, you had general symptoms that you would get with any kind of illness. But for a period of illness now, you have specific symptoms. So for instance, if it's chickenpox that you have, now you'll get all of those red spots across your body. If it's an enteric infection, meaning your gastrointestinal, well, now the specific symptoms will be the vomiting or diarrhea. Okay, so now the period of illness, not only do I want you to remember that it's specific symptoms now, but I also want you to write down that this is when you may die. Okay, so if someone's going to die from an infection, it's during the period of illness. Then you hit the period of decline. The period of decline is, as it suggests, the symptoms are declining or decreasing. Okay, but I want you to put a star next to the period of decline and write that even though you start to feel better, this is when you can acquire secondary infection. Okay, so for period of decline, write down that you can acquire a secondary infection. Okay, why is that? Because think about it, at this point, now you have become immunocompromised, right? So if you're immunocompromised, what can happen? Opportunistic pathogens. So now that you're weakened, now your normal flora, who are usually the good guys, they can jump in and make you sick. Okay, you can also, because you're in a weakened immune state now, you can also pick up infections in the outside world where you may not have normally gotten sick from these things because, you know, you were strong enough before to fight them before you really saw any illness. So that's why I always tell my students, be very careful when you're just getting over an infection. So even things like the cold or flu. Because when you're starting to feel better, you're very, very susceptible to secondary infections. And a lot of times people will say, oh yeah, I relapsed because I was running around too much after I was getting better. It's not that you're relapsing from the same infection. It's that you're actually picking up a secondary infection. So please, anytime you're recovering from being sick, okay, even though you're starting to feel better, do not rush back to places like stores, the gym, or school, and even work, okay? Please try to avoid as many public places as you can while you are starting to feel better from an infection, because you very easily will pick up a secondary infection if you're not careful. And if you don't pick up a secondary infection, then hopefully you make it to the period of convalescence. And that simply means you're now all better, you beat it, and you've regained your strength and immunity pretty well by this point, okay? So now whenever you hear the term convalescence, just know that's a fancy way of saying you feel better. So if anyone asks you how you're doing, you say, I've convalesced, thankfully, okay? So again, make sure you're very comfortable with the 5 stages of infection, not only their name, but the significant aspect of them. So for instance, incubation, no signs or symptoms yet. Prodromal, general symptoms. Period of illness, specific symptoms, and you can die. Period of decline, feeling better, but can pick up secondary infection. And period of convalescence, you are back to normal, healthy and good. Ugh, I know, so much terminology in this lecture. Well, here's another slide full of terminology, and once again, these are all terms that I'm sure you've heard before, but we need to make sure that you're truly clear on how to distinguish them. So this slide, we're going to start looking at the duration and scope of disease, which means how long does a particular infection last, and how far does it spread? So when we talk about duration, there's a few different categories. An infection can be acute, chronic, subacute, or latent. Acute means any of the infections where the symptoms you'll notice will develop really quickly, but the disease itself will only last a very short time. So things like if you think about having a cold or the flu, you know, they'll hit you very quickly, but they're not going to last very long and you'll start to feel better. Chronic, on the other hand, the symptoms you may not even notice at first are starting to build up. The symptoms develop very slowly in chronic illness and then last for a very long time. In fact, chronic illness can a lot of times even be lifelong. Okay, the big example of chronic illness would be things like TB or lupus. You then have infections that are subacute, and these are rarer to hear. Subacute infections means that they're basically an intermediate between acute and chronic. So with these infections, they'll develop slowly the way a chronic illness would, but they can kill very quickly the way an acute, very quick, short illness would. Okay, so with subacute, again, think of slowly developing, but then kill quickly once active. And a big example of this would be asthma. So for instance, asthma may progress very slowly, but if a child has a sudden asthma attack, it can kill them very quickly. The last one with regard to duration are the latent infections. So for instance, a lot of STDs go latent. Now latent, what that means is it's another word for dormant, okay, dormant or inactive. Now latent infections, what you notice with these is the causative agent, whatever microbe is causing this infection, goes inactive for quite a bit of time, but then will constantly reactivate and produce symptoms again. So big examples are things like herpes or shingles. Okay, and whenever you encounter or deal with any of the latent infections, the two things that tend to really make them reemerge or reactivate, one is if you end up immunocompromised, so kind of like when you have opportunistic pathogens as well. So being immunocompromised will reactivate latent infections, and times of stress. Okay, the more stressed out you are, the more likely these latent infections will pop up, which is why you'll notice that people who have, let's say, latent herpes or even mono, what'll happen is during times like finals week, suddenly you'll see the blisters kind of popping up and cold sores popping up, okay? Now, in addition to duration, how long these symptoms are occurring, you also have the scope of infections. And what that means is how widespread on your body the infection is. So local is exactly what it sounds like. Local means the pathogen stays at the original location, so the infection is just at one spot. In fact, we usually call that spot the focus of infection. So for instance, if you get a splinter and you just have, that splinter in your finger, and your finger, that one spot is red, swelling up, painful. Okay, that would be a local infection. Whereas if that infection then spreads throughout your blood and your lymph and spreads to other organs, well, now that's systemic. Okay, so local is one spot. Systemic is traveling through your body. So blood and lymph is now included. There's some terminology with systemic infections that you have to recognize or be familiar with. The first one is septicemia. And what septicemia is that there are pathogens or microbes in the blood, whereas toxemia is specific to toxins in the blood. You then see two different forms of sepsis listed. And talking about systemic or sepsis infections is very important, especially if any of you plan on going into the medical field, or if any of you or your loved ones end up in the hospital, because I want to point out that over 250,000 Americans die each year from sepsis, including my own father. So I like to always point out and emphasize to all of the students, especially those of you going into the medical field, to be careful, because systemic infections are becoming more and more common, especially in hospitals. Okay, and the two forms of sepsis that you see on this slide are severe sepsis versus acute sepsis. And when you see these terms, whenever you hear acute, based on what we were talking about a minute ago, acute means very quick. It all happens very quickly. So with acute septic shock, your patient can suddenly die within 24 to 48 hours because there's such widespread tissue and cell damage. Whereas with severe sepsis, they may have a couple of weeks before they're killed if you don't get to really culture and identify exactly what's causing that sepsis infection. Okay, so in severe sepsis, you still get the systemic inflammation and organ dysfunction, but it's at a lower rate, so there's still more time to try and save the patient. Where again, acute septic shock, within 24 to 48 hours, your patient can die. Okay, so make sure you're very comfortable with the differences amongst the various terms on the slide. And to help solidify your understanding of them, here's a recap of the notes again to write down, just in case I went too quickly over any of the terms we discussed. Okay, so once again, anytime you see a recap note slide, just simply pause the recording and make sure to write down all of the terms that you see. Now, the next thing to cover in this lecture is epidemiology. Whenever you hear epidemiology, that simply means the study of factors and mechanisms involved in the frequency and spread of illness. Okay, so basically we're looking into frequencies, rates, things like, how many cases of an infection are you seeing? Okay, there's a lot of terminology here you have to be comfortable with, and especially if you go in the medical field, you'll have meetings dedicated to epidemiology and looking at things like incidence, prevalence, morbidity, mortality. Okay, so our terminology, first one is incidence of a disease. So incidence would be the number of new cases that you're seeing pop up for a particular disease, and it's always given in a particular population and a particular period of time. So for instance, within 6 months in, Lakewood, we saw 200 new cases of whooping cough. And so we would say that the incidence of whooping cough in this location during that time would be 200. Now, if instead you are asked for a rate, then what you do is you put the number of cases over the total. So anytime you hear the term rate, then you should be giving a fraction as an answer. So in that same scenario for whooping cough, where I said 200 new cases in Lakewood during a 6-month period, the incidence would be 200. But the incidence rate would be that 200 over the population. So let's say we said the population was 100,000. Well, the incidence rate would be 200 over 100,000. The next terminology is prevalence. Okay, prevalence now, instead of just the new cases, this is the total number of people with that infection. So that means any of the new cases plus anyone who already had this infection. So for instance, if we said that those 200 new cases were on top of 500 already sick people with whooping cough, then the prevalence would be 200 plus the already 500 that had it. So that would be 700 as the prevalence. Okay, so the prevalence would be 700. And if instead I said prevalence rate, then once again you would simply put that 700 over the total population, which I had said in the example was 100,000 people. Okay, the next one is morbidity rate. And morbidity rate is simply another way of saying prevalence rate. Okay, so next to morbidity rate, you can write prevalence rate. Okay, because it means you're taking the total number of people with the disease, so that's new and existing, divided by the total population. Okay, so I'll never have you solve for it. You'll just have to write out the fraction. So for instance, morbidity rate in the example that I've been giving would have been the same answer that I said for prevalence rate. It would be 700 over 100,000, which was the total population. The last term to know is mortality rate, and please do not get the terms morbidity and mortality mixed up, okay, because that's a very common mistake, but they're two different things. Mortality rate is simply the number of deaths divided by total population. Okay, so if I said the mortality of the infection, it would be how many people died from it, whereas mortality rate is taking into account the total population. Now, when we talk about mortality rate, I want to point out that the total population never changes for that set time. Okay, you do not deduct the people who died. So, for instance, if I said that during that time that we were just talking about, 6 patients died from whooping cough in that population, we would say 6 over 100,000 is the mortality rate. Okay, notice we did not change the 100,000. Now, the best way to really understand these terms is to practice questions with them. So now on the next slide, I'll have an example, which is the exact format that you would find these questions on an exam or quiz. So here we have an example question, and like we've done in other lectures, you have to send me your answers to these questions in Remind by Saturday at noon. Okay, now what this question asks is it first gives you a scenario. It will give you a time period. It will give you information about a particular population. You then have to figure out things such as incidence rate, prevalence, and so forth. So I'll always first tell you a time period. So in this case, between January to June 2016, 110 new cases of whooping cough, pertussis, appeared in Tardisville with a population of 12,000. This brings the total number of citizens with whooping cough up to 212. Unfortunately, 5 people died from the disease during this time span. I want to point out that the other way that I can ask this, so instead of giving you the total number of citizens, I could have said that the 110 new cases were added to 102 people who already had the infection. So always read the question very carefully to see whether I tell you new and total or whether I tell you new and existing and you then have to add together the new and existing to find the total. Okay, so please make sure that you take a look at these 5 questions and send me the answers in Remind by Saturday noon as part of your participation in Remind grade for the total course. If you're having any difficulties, as always, just send me a message either through Remind or through email, and I will help you to guide through this question. The next thing you sometimes encounter when we're talking about things like epidemiology are the terms endemic, epidemic, pandemic, and also sporadic. Okay? If a disease is endemic, what that means is it's constantly present in a population. So the example I always give for that is malaria in Africa. Okay, so this disease is always present at a rather large amount. You then have in this figure sporadic, and as you notice, sporadic shows just every once in a while a little bit of a pop-up of new cases for a particular disease. So sporadic means only occasionally are you seeing new cases pop up of this infection. Then we have the terms epidemic and pandemic. And unfortunately, things like the news or the media have a tendency to mix and match these terms, even though they have 2 very different meanings. So if something is an epidemic, this means that the new cases of the disease are acquired by many people in a given area in a short time, meaning one location, one region, suddenly has a large outbreak of new cases of a disease. Pandemic, on the other hand, if you notice here, this is now multiple regions. It is a worldwide epidemic. So for pandemic, what I usually do is I underline the P of pandemic, and I think of the singer Pitbull, Mr. Worldwide. So pandemic is when the new cases are popping up worldwide. And a big reason for pandemics nowadays, travel. Think about the airline industry. That opened up the whole world for infections to spread very easily, very far. Okay, so pandemics, a big part of it is thanks to travel. So now, once again, to recap, in case you had any difficulty writing down what I was saying, if I went a little too fast or anything, the 4 definitions that we had from the previous slide are sporadic, meaning only occasionally do you see that disease. Endemic, meaning the disease is constantly present. Epidemic, meaning suddenly in a short time in one location you have the disease popping up. And pandemic is when the epidemic has now gone worldwide. So once again, feel free to pause the recording in order to write all of these notes down. So speaking of pandemics and the spread of disease, when you think of the spread of disease, you can think either big worldwide spread or even within your own home, your own everyday life. Okay, there's various ways for disease to spread because there are various reservoirs of infection, meaning things housing the pathogens. So the first big category of reservoirs of infections are humans. Okay, and humans as reservoirs can either be sick people or they can be carriers. Okay, so what's the difference between sick and carriers? Well, are you seeing the symptoms, which is a sick person, or does it not even look like they're sick, but they are incubating that infection, which can still spread, and those are carriers. The other reservoir option is animals. And when an animal transmits pathogens, we call those zoonotic diseases. So think of the zoo, animals. Zoonotic diseases are when animals are carrying the pathogens. And you can get zoonotic diseases in more than one way. Okay, you can directly get them from things like, an animal bite, or you could get them transmitted from things like fecal matter. Okay. The next one are the non-living reservoirs, and these include things like water, soil, food, air. Okay. And when you look at all the reservoirs on this slide, from humans to animals to non-living reservoirs, including water, soil, food, you have to ask yourself, which of these reservoirs would you consider the most dangerous? And out of all of these reservoirs, you may be surprised to know, water is the most dangerous of all the reservoirs. Because think about it, you cannot live without water. You can't find any alternative if the water is contaminated. Okay? Humans, yeah, sure, you can avoid people. I do it all the time. You can avoid animals. I know some people who are terrified of things like dogs, and they will run across the street just to not walk anywhere near a dog. And non-living reservoirs, well, things like food. Okay, so on the news they say romaine has been, contaminated. Okay, I just won't eat a salad. Still plenty of other options to eat. Whereas water, you can't avoid water. Okay, so water we consider the most dangerous of the reservoirs. And contaminated water will cause infections like typhoid fever, cholera, okay, diarrhea, things that we talk about a lot in the lab when we do water control, quality control testing. Now, when you think of the spread of the disease from those reservoirs, there are various mechanisms that the infection can be transmitted. The first one is contact transmission. And when we talk about contact transmission, that means that you have either touch or close contact, okay? And contact transmission can be broken down into 3 different types of contact. There can be direct, indirect, or droplet contact transmission. So for direct contact transmission, that would be things like touching, kissing, biting, sex, any of those things, okay? Where you are directly contacting the sick individual, okay? Indirect, however, now you have a non-living intermediate, okay, to the sick individual. Now notice with indirect, okay, the non-living intermediate will be things that you associate with a sick individual. So for instance, needles, used tissues, hospital bedding, anything that you would have seen a sick person using, okay, that's indirect. Then you have droplet, and droplets would be if the sick individual sneezes, coughs, even laughs, and you get those little saliva airborne droplets. Now, when we talk about droplets, you should always ask yourself, what's more dangerous, smaller droplets or larger droplets? The answer is smaller droplets. They're more dangerous because if you think about it, if it's a small droplet, it could be airborne longer and further than a big, heavy droplet. Okay? The next example of transmission is vehicle transmission. This is the one where pathogens now are hitching a ride on something you would think would be clean, okay? This would be something such as food or a fomite, like we talked about in lab. So in the case of vehicle transmission, the reason why it differs from indirect transmission, in vehicle transmission, the object is something that you don't associate with a sick person. Instead, it's an object, a fomite, that you would think is perfectly normal for anyone. Things like a doorknob, cell phone, glasses, your ring, your watch, okay, a shoe, any of these things that it's not like you see a sick person coughing and sneezing all over it the way you would with a tissue or a medical needle, okay? The last type of transmission is vector transmission. And vector transmission can either be mechanical or biological. Either way, a vector is a carrier organism, usually an arthropod, or something such as a fly, a tick, a mosquito, any type of bug that you would think of. Okay? When we then categorize it as mechanical or biological, there are two ways that this bug can get you sick. In mechanical is physical movement, right? So mechanical vector transmission would be like if you see a fly land on your food and suddenly its body parts are contaminating your food. Okay, so it's brushing off with its little legs, looking like a little villain, rubbing its legs together. It might be brushing off pathogens onto your food that you then eat and get sick. So that would be mechanical, when their body parts are contaminated and make you sick. Whereas biological, what's the other way an insect can make you sick? Well, by biting you. Their biological fluid. Okay, so mechanical is their body parts contaminating you, whereas biological transmission is biological fluid from a bite. So things like a flea bite causing the plague, or a mosquito bite causing Zika, West Nile, malaria. Any of those would be biological vector transmission. So again, just to recap notes, because there was a lot of information there, I wrote down the key points, and you can once again pause the video in order to write down all of the notes. Now, just to help you visualize what we were talking about, we're going to go through each of the transmissions in picture form. This first form is contact transmission, where the first one you see is direct contact, where you kiss or bite or have sexual contact with the sick person. Then you have indirect contact transmission, which is inanimate objects that the sick person has touched and that you associate with sickness. And then you have droplet transmission, which are the bodily fluids that are exposed through sneezing, coughing, things like laughing. Vehicle transmission, we have the examples of water, food, air, and also you may want to remind yourself fomites, okay, F-O-M-I-T-E, which are the inanimate objects such as a cell phone or a doorknob handle. Then we have mechanical and biological vector transmission. Again, the fly on food, that would be mechanical, whereas biting into your skin, that would be biological. Now, as we talk about the spread of infections, one of the biggest places where infections spread is actually the place you go to try and make yourself feel better after an infection. So it's actually the healthcare locations, such as hospitals or clinics or doctor's offices, where you actually tend to pick up the most infections. As we've mentioned before, we tend to call these nosocomial infections, meaning any infection picked up in a healthcare facility, okay, affects 1 in 25 patients. And as I said, you know, the amount of sepsis in hospitals is jumping every year. And they result from things such as, you know, the microbes just being present in a hospital environment, because that's where sick people go. Also, the fact that a lot of people there, since they're sick or dealing with recoveries, they're compromised. So we said immunocompromised patients end up being the most likely to pick up infections. And the last thing here is the idea of the chain of transmission, which in the medical field, we're constantly trying to break that chain. So the figure over here demonstrates an example of the chain of transmission in a hospital. You have the pathogens which are currently in a reservoir, such as the doctor. That reservoir then has a portal of exit when he coughs into his hand. He then goes into a patient's room where he shakes the patient's hand, and that is direct contact transmission, without realizing that they have a cut on their fingers, and so you have a portal of entry, and the susceptible host, who is currently immunocompromised, quickly gets that infection from the doctor, okay? So when you're working in the medical field or when you're visiting loved ones, be very, very aware of the chain of transmission and try to avoid any unnecessary contact or any, how would we call these, maybe naive actions, okay? So basically meaning anything that you may not normally think of, like, oh yeah, my nose is a little, having some sniffles, let me wipe my nose with my hand. Well, not in the hospital, okay? So be very aware of your actions and always picture microbes and how they are getting spread. Here we kind of summarize some various ways that can help control these various infections, these hospital-acquired infections. Okay, you want to reduce the number of pathogens as often as possible. So make sure to hand wash regularly. Use things like disinfecting the disinfectants on tubs or equipment. Okay, make sure to clean any medical instruments very, very carefully and very frequently. And when possible, try to use disposable items, okay? Because disposable items are kept sterile until you use them and then thrown away. So you reduce the risk of spreading any kind of microbes to other patients. Because sometimes, no matter how much we try to clean instruments, there are always going to be some highly resistant infections, okay? And also, another thing that they try to do to help control these infections is by having control committees that really strictly regulate all of the actions within a medical facility. But again, your education and awareness are going to be the biggest things to control these infections. Be very aware of everything you're doing, and please be very careful of the patients, but also be careful for yourselves and your loved ones, because you never want to track any of these pathogens you encounter back home to your loved ones. Okay, so that's it for today. Make sure to head over to the discussion board and contact me and Remind if you have any questions or concerns.